Good morning once again. Welcome to today's webinar, Nassau Grupa, the Heart of Bahamian Fisheries. Our speaker today is Dr. Krista Sherman. She has been studying this iconic species for over a decade. Thank you for joining us today to learn more about her work and the crucial efforts to safeguard the species from extinction. So I'll share a bit of Dr. Sherman's background. Dr. Krista Danielle Sherman is a marine scientist with a broad range of research and conservation experience. She holds a PhD in biological sciences, a Master of Research in ocean science, and a Bachelor's of Science in marine science and a minor in Spanish. Her PhD research assessed the status, population structure, and dynamics of Nassau Grupa spawning aggregations, was critical to the development of the first Nassau Grupa Conservation Management Plan for the Bahamas as well. Dr. Sherman is currently a senior scientist for the Perry Institute for Marine Science and leads the Fisheries Research and Conservation Program. So just a few housekeeping rules before we begin. Please note that this webinar is being recorded, and a reminder to keep your microphone muted during the session. Afterwards, there will be a Q&A session. Add any questions into the chat or raise your hand during the Q&A session. An audience feedback link will be shared in the chat. So without further ado, I will now welcome our speaker to begin their presentation.
Thank you for the introduction, Danielle. Good morning, everybody. Thank you for joining. So for today's webinar, I'm going to be talking about some of our past and ongoing research efforts focused on Nassau grouper and how this is being applied to help conserve this species. It's really important. So before I get started, I'll tell you a little bit more about my background. I was born and raised on the island of New Providence, and I was really fortunate to have been introduced to the ocean as a baby. I loved it then, I still do, and that love, as well as the curiosity about marine life and how everything worked and love for science, is really what inspired me to become a marine scientist. And I've been doing this for about 16 years now. In 2018, I joined the Perry Institute for Marine Science, and PIMS is a nonprofit research and conservation organization that's really been at the forefront of trying to address some of the issues that are impacting the marine environment. So for the last 5 decades and more now, really using science as a foundation to guide and inform conservation policies and practices and engaging people in that process. As Danielle mentioned, I lead our Fisheries Research and Conservation Program, and I've been working locally with scientists, internationally with scientists, and other key stakeholders to address some of the important gaps that we have for marine species. I work with both traditional fishery species, like we're talking about today, the Nassau grouper, other reef fish, queen conch, but I also work on emerging fisheries as well. So sea cucumber, parrotfish, re-emerging fisheries like the sponge fishery. And the goal of all this work is really to promote sustainable fisheries and to ensure that people can continue to benefit from them. So the objectives of this program are to reduce some of the knowledge gaps that we have for these important marine species, to build capacity, to develop science-based management plans, to provide guidance for fishery regulations, marine spatial planning, and ecosystem-based management more broadly, and to really promote behavior change in the Bahamas and the Caribbean so that we have improved conservation management. So working on public awareness. And that's something that we're going to talk about today with a focus on Nassau grouper. Let me see if I can get this to disappear and not be so prevalent. Okay. So Nassau grouper are really important teleosts or bony fish predators on coral reef habitats. And they're also an important food source for larger apex predators. They are considered to be a K-selected species, so they have long lifespans, about 29 years, slow growth rates, and delayed sexual maturity. But if allowed, this fish can get really big and grow up to about 4 feet in total length from snout to tail. and weigh up to 60 pounds. So that's pretty impressive for a fish. They are native throughout the tropical and western Atlantic. That includes Bermuda, Florida, throughout the Bahamas, the Caribbean Sea, and parts of the Gulf of Mexico in marine habitats to a depth of about 255 meters. And we care about this species because in addition to their ecological importance as predators, the formation of their spawning aggregations also releases important nutrients that are important for reef health, but they're also highly valued economically and culturally in the Bahamas and throughout the region. So we really do care about this species. Nassau grouper, like other reef fish, have what we refer to as a bipartite or a two-phase life cycle. So starting off in the water column as larvae, and they're subject to being moved around based on ocean currents at this stage. But when they're ready to recruit or settle into nearshore nursery habitats, they settle into these areas at about 1.5 to 2 inches. And it's a balance at this point of being able to find shelter and avoid being eaten, because natural mortality, as you can imagine, is pretty high in these early life stages, but they also need to be able to grow. And they gradually transition and mature and move out to other habitats, so to patch reefs as juveniles and subadults, and then the adults, which attain maturity between the ages of 4 to 8, then transition on to more structured coral reef habitats as we know them. And it's at this point that they're ready to begin this process, this life cycle all over again. And it's really, really interesting because Nassau grouper have a really unique way of reproducing, and that's through the formation of fish spawning aggregations, or FSAs. So for those of you that aren't familiar, I'll just explain this a little bit. But essentially, it's where you have repeated concentrations of the same types of marine animals, and they're gathered there in order to breed or reproduce or spawn. More importantly, when this occurs and where this occurs is quite predictable. In addition to that, the densities of fish that are participating at these aggregation events are 4 times greater than what you would find outside of the aggregation. So you have a lot of offspring, a lot of gametes, eggs, and sperm being released into the water column at this time. The actual first spawning aggregation that's documented through the region actually occurred here in the Bahamas. That was back in the early 70s, and it was published in 1972. And they estimated up to 100,000 fish at one single spawning aggregation site during that period. And the author at the time, the scientist that was there, Smith, noted that if these sites were destroyed because of unsustainable fishing practices, or if there was a significant loss to their habitat, that there would be a reduction in terms of the populations. And that's something that we wouldn't notice immediately because these are such long-lived fish. And that is something that's actually happened. This spawning aggregation site no longer forms. What's happening? Sorry, having some technical difficulties here. Got it. Okay. Okay. So for those of you that aren't familiar, this is what it looks like underwater. So you have these hundreds of thousands of fish gathered at these spawning sites in order to breed, in order to reproduce. Nassau grouper transition in and out of these different color phases. Towards sunset, they'll break off in smaller groups. They'll be nudging, chasing, engaged in these incredible courtship displays, and then will release their eggs and sperm into the water column. And we believe this reproductive strategy evolved to maximize reproductive success because outside of the spawning season, Nassau grouper are quite solitary. So it makes sense for this species to reproduce in this way. But because when these spawning aggregations occur and where they occur are really predictable. They're often well known to fishermen. And this has led to a lot of targeted fishing during the times when they're most vulnerable at these spawning aggregation sites. And this is something that's not often realized because the fish are concentrated, you're still able to relatively catch the same densities of fish over time, but until the point where that doesn't happen anymore, but the population size is also declining. So it's a phenomenon that we refer to as hyperstability. And this is really problematic for a number of reasons. Obviously, we're decreasing the size of what we call the big old fat fecund fishes, or the BOFs. So we're not seeing replenishment of the fishery. That has influences and can impair actual courtship and mating behavior. Obviously, reduced numbers of fish at these spawning aggregation sites means that any predation on the fish themselves or the offspring becomes more problematic. We often see large bull sharks at some of these spawning aggregation sites. On one expedition, a whale shark was observed. So this is problematic. Sorry, this is still getting in the way, and leads to potential declines in the population, which can impair and influence genetic diversity. So Nassau grouper globally have declined by 80% or more. And we've seen FSA collapses, not just here in the Bahamas, but throughout the region. And this is what led to the IUCN classifying them as critically endangered. They've also been listed on the Endangered Species Act as threatened and are on Appendix 3 of the SPA protocol because of their extreme susceptibility to being overexploited. And we're interested in pursuing this work in the Bahamas for a number of reasons. One of them is that the Bahamas has a lot of the essential habitats required by Nassau grouper throughout their life cycle, but we also have the largest number of reported spawning aggregation sites compared to any other country where spawning aggregations are historically reported to occur. In addition to that, Nassau grouper are an economically important species in the Bahamas, one of the most commercially important and valuable fish species. But even with these commercial landings, we've seen declines over the last 2 decades, and that's cause for concern because it's something that supports fishers as well. What is happening? Sorry, guys. Some of you may have heard or watched the news, looked at the news this week, and noticed that there's still a demand, even though we have an established closed season for Nassau grouper during the months when they're breeding. And this was one of the headlines from the papers this week. So their life history coupled with this continued demand is really driving declines in populations of this species. So one of the first things I wanted to figure out is, this is happening, what do we need to know, and how is this impacting grouper populations? So some of the questions that we wanted to address were how genetically diverse and connected are Nassau grouper populations in the Bahamas? What are the actual demographics, the spawning stock sizes, the predominant migration patterns of the remaining spawning aggregation sites that we do have? How is overfishing impacting the reproductive potential and success of this species? And how should we adapt our marine protected area networks, add to that, refine that to better account for connectivity and to promote recovery for this species? So at PIMS, we've been using a range of techniques and methods to study Nassau grouper over the last, well, I guess more than 10 years now. I'm only going to talk about 2 today, but I'll highlight some of the other ones. So obviously the in-water assessments that you've seen little snippets from, where we're looking at abundances, courtship behavior, documenting any spawning events and fishing activity that's happening there. We also work with a tagging and acoustic telemetry, which I'll talk about more today. We've pilot tested passive acoustic monitoring, thanks to collaborations with Dr. Michelle Scherer from the University of Puerto Rico and HJR Reefscaping, as well as the Bahamas Marine Mammal Research Organization, in addition to collecting video surveys. The underwater visual surveys, I should mention, are not just at the spawning aggregation sites, but that's something that we do throughout the Bahamas and other habitats that Nassau Grouper uses. So doing reef assessments, also doing mangrove assessments as well. And then obviously we've done some genetics work and then collaborating with Lindy Knowles from the Bahamas National Trust on habitat and bathymetry mapping. So just want to share, again, an underwater clip of what some of these spawning aggregation sites looks like. It's really incredible to be under there, to be a part of that, but it's one of the most difficult things that I do field work wise, because spawning aggregations happen to occur in the winter. I don't know if any of you have been outside today, but it's it's pretty rough. So it makes it challenging. A lot of the sites that we go to are remote, and there's no access to dive shops that have tanks, so we need to charter liverboards. So it's really, really expensive to do that work as well. So, you know, this work has not been consistent over the years because of the costs and because of the weather, making it extremely challenging to do. Of the up to 40 spawning aggregation sites reported in the Bahamas, we're only making a dent. There's a lot of areas that you can see here that are still unknown, and those are in the gray little pentagon looking things. But we've been revisiting as we can and are able to some of these spawning aggregation sites that have been reported to us by fishermen to verify whether they're active or not for Nassau Grouper. A lot of the ones that we have been to and revisited since 2010 have reduced numbers of fish compared to what they were reported historically. So we've done some work around Long Island, in Andros and the Berry Islands, off of Key Verde and Ragged Islands, where one of the largest spawning aggregations are. Done some work around parts of Eleuthera as well, in 2019 in San Salvador with a local fisherman. And the last time we were in the water doing assessments was 4 years ago in Abaco. So this is what we've done to date. There's still a lot of unknowns. There's still a lot more exploration that needs to be done in terms of verifying whether these sites are active or not for Nassau Grouper and when. But for the first, or I guess the next part of this talk, I'll talk a little bit about acoustic telemetry and tagging, which essentially involves us being able to track movement patterns of fish to and from the reefs to these spawning aggregation sites. The first study in the Bahamas using this technique was led by Dr. Craig Dahlgren, and he tracked 19 fish over the course of 3 years that were tagged from the Genomic Land and Sea Park with these acoustic transmitters. So they're internal tags. You have to surgically implant these within the fish, and that information gets detected once the fish pass within range of the receivers that you see here on the screen here, and recorded, so the date and time. And that information showed that all of the fish migrating were at least 54 centimeters or bigger, which is bigger than our current legal size limit, which is based on the age of first sexual maturity from the region and age at which fish are migrating in other parts of the region for the first time. But that's not what's happening here in the Bahamas. And what he found is that all but one fish left the protection of the Genomic Land and Sea Park, which is a no-take marine protected area, and went down to Long Island here. But they're using different spawning aggregation sites, which are in these pink circles. So in some instances, they've bypassed a spawning aggregation site, which is closer, to travel all the way down to Long Island. His work also revealed that these fish are doing this in groups, so they're making group migrations. They migrate out from their reefs, where they're normally solitary, out to the shelf edge in groups, and then they go to their preferred spawning aggregation sites. So there's some site fidelity there. And that really varies depending on the timing of the full moon, which is something that was also quite interesting from that work. The second study using this method was focused on the tongue of the ocean system, including Berry Islands and Andros. We had an array of receivers set up, And again, this kind of confirmed and was consistent with some of Craig's work in terms of fish making these long distance migrations, sometimes round trip over 260 kilometers to get to a preferred spawning aggregation site. But the interesting thing from this study is that this work showed that an historic spawning aggregation site, one of the first to receive protection for the Bahamas, High Key, appears to have collapsed and is no longer forming, and fish are bypassing that to go to a spawning site in the northern part of the barrier, northern part of the barrier reef. So that was from this study that was led by Christine Stump from the Shedd Aquarium. Since then, I've also, and more recently, established an array of receivers around San Salvador, since we were not able to verify any spawning using the in-water assessments with the guidance of local fishermen back in 2019. Don't have anything to report. This is work that's still ongoing. So hopefully in a couple of years we can share an update on what's happening in San Salvador, and I think that would be really interesting because it's isolated from the Great Bahama Bank and the Little Bahama Bank, where we know other spawning aggregations are. So there must be something happening around here, and there's some likely good spots where fish may be aggregating. So stay tuned. But acoustic telemetry also has a cost. You know, each of those little transmitters is several $100s. Each of those receivers is over $1,200. So obviously we can't tag thousands of fish. It's too expensive, it's too costly. But fish have their own built-in tags, their DNA, if we know how to use them. And so population genetics can be a really useful technique if we know which markers to use to help answer some really important questions that can guide fisheries management. And I use this technique because there had been some previous work, population genetics work done in the Caribbean, but it was reduced both in representation and sample size and spatial scale from the Bahamas in this regional study that looked at genetic differentiation in the Caribbean, which suggested that the Bahamas Nassau grouper populations may be distinct from the rest of the Caribbean. But as you've seen this graph before, we have a lot of FSAs, a whole lot more area to cover, and we knew really very little about genetic connectivity of Nassau grouper. So for the purposes of this work, we wanted to determine how genetically diverse and connected Nassau grouper populations within the Bahamas were, and to explore whether human impacts or anthropogenic activities have actually compromised the health of these fish, looking at a metric called effective population size. And so that's what I'm going to talk about now. So over the course of 3 years, and in collaboration with partners in the country, collected over 464 fin clip samples for genetic work. So we covered 13 islands from the northern extremes and southern extremes of the Bahamas. And in addition to sampling fish over 3 consecutive spawning aggregation seasons from this Hail Mary spawning aggregation site. And for the first component of this work, I used microsatellite genotyping. And microsatellites are a commonly used molecular marker for population genetic studies, and really use this technique to understand what the genetic architecture and current demographics of Nassau grouper are, and to detect whether any changes have occurred to their population structure. So what you're looking at here is what's called a structure plot, and it's organized geographically from north to south. And the Hail Mary spawning aggregation site is right here. So each of these bars correspond to a single or an individual fish, and the genotypes are represented by 2 different colors. So in this case, aqua and dark blue. So what you're seeing is that some fish have more of one genotype than the other. And overall for the Bahamas, based on this type of analysis, there appear to be 2 genetic stocks. But the important thing here that there's genetic mixing throughout the country. There's no geographic population structure. So the next thing we did was to run some analysis to examine how the numbers of Nassau grouper contributing to the population size have changed over time, and compared that to historic levels from the present to 1,000 generations ago. And what we found is that there was a systematic crash in population sizes all throughout the Bahamas. So the Bahamas overall, and for all the different locations that we sampled, including the Hail Mary spawning aggregation site in green. And this decline really began around 400 generations ago. And there was an accelerated decline within the last 150 to 200 generations. And the important thing here is the timing of this decline was about 3,600 years ago, and it predates any anthropogenic or human fishing activity. So that suggests that natural processes were responsible for historic declines in Nassau grouper populations in the Bahamas. But we also looked at contemporary estimates, what's happening more recently in recent times. And we see that there's also evidence for significant reductions in population size throughout the Bahamas, with bottlenecks occurring in a couple different islands, Abaco, Eleuthera, and Exuma, as well as this Hail Mary spawning aggregation site. So what the genetic data show from the microsat analysis are that Nassau grouper are experiencing reductions in the numbers of breeding individuals that are contributing to successive generations, and also these bottlenecking events, which have implications for their long-term ability to adapt to stressors. So that was the first phase of the genetics work I did. The second phase was to use genomic markers, and that's because they cover a whole lot more of the genome than microsatellites. So keep your eye on the Hail Mary at the bottom of the right screen. So we expected by using genomic markers like based on RADseq that we can improve the resolution and capacity to unravel some of the genetic diversity and divergence that we're seeing in Nassau grouper, despite the fact that with the microsatellite information, they're showing weak population structure. So genomic techniques allow us to have better resolution into what's happening. So I used RAD sequencing to establish countrywide demographic structure and for genome-wide assessments of diversity and differentiation. So to do this, I used the same samples that I collected previously, a subset of individuals, and extracted high molecular weight DNA from 96 fish. From the islands that you see here on the screen in blue, as well as a Hail Mary spawning aggregation. So what you're seeing here is analysis based on that information, and you can see that there's a distinct separation between Exuma and this light aqua color here and Long Island from the rest of the Bahamas, including the Hail Mary spawning aggregation site. And this is just a map here showing you a reference with where those things are. So because geography is clearly not driving the patterns of differentiation that we're seeing, we tried to explore whether selection may be influencing the patterns that we observed. So we looked at environmental association tests to screen for loci that may be under adaptation or selection. And used this to visualize whether patterns of genetic structure could be attributed to these loci that may be under selective processes. So what you're seeing is that outlier SNPs from these tests, we align them to a genome that was developed for red spotted grouper to look at some of the functional attributes. And they were connected to a range of important biological functions. But you can see that in the northern part of the Bahamas, current and sea surface temperature appear to be important. And then the southern part of the Bahamas and Ragged Island here, primary productivity appears to be important. So without having physical barriers to gene flow, it's really interesting that we're seeing this distinct separation between Exuma and Long Island. And so there are probably a few explanations that seem likely why we're seeing the patterns that we're seeing. And this includes the fact that these adults are making these really long distance migrations, and it's really varied with site fidelity as well. There's also a lot of variability we know in actual larval survivalship. Mortality is high in those stages, but it's quite variable in terms of recruitment and how that's dispersed, and that gets carried on and reflected throughout the population based on some earlier work that's been done. We also know that because fishing pressure remains high, there are lower densities of fish outside of the Exuma Keys Land and Sea Park than there are inside. And then there may be just different selective pressures at play here. And because fish are making these long distance migrations, there's probably a cost associated with this. And so it may be energetically more demanding. And so that's perhaps why we're seeing some of these outlier SNPs associated with osmoregulation, and with metabolic processes and muscle function, which were also linked to the environmental variables that I showed you earlier. So the key findings from both the telemetry and genetic work, we'll start with the telemetry work. Fish are making group migrations. So it's possible to collect fish when they're on these migratory corridors to and from the spawning aggregation sites, in addition to overharvesting them at the spawning aggregation sites. These long distance migrations can be over 250 kilometers, so longer than Long Island, for example. And there are differences in the timing of when this happens, linked to the full moon, but also differences in swimming speeds. More importantly, fish are migrating for the first time, most fish on average, when they're 54 centimeters or bigger in total length, which is bigger than our current legal size limit in the Bahamas of about 3 pounds or about 48 centimeters. In terms of genetic diversity, that was from the microsatellite information that was similar, but low allelic richness. So again, compromised ability to adapt to stressors moving forward. There is a bit of population structure with clear separation between Exuma and Long Island, and a lot of work in the future, I think, is warranted to try and figure out why that is. But overall, these fish do have impaired genetic health based on the metrics that we looked at, both infective population size and bottlenecks as well, that have occurred historically before humans were at play, so things that have happened in nature prior to that, but also more recently because of the intense fishing pressure on some of these spawning aggregation sites. So if you would like to read more about any of this work, the papers are online. I think most of them are open access, but if not, feel free to shoot me an email and I can send those to you. So how is this information then being applied to help with the conservation of this fish? So that's what we're going to talk about now. In addition to all of the actual field work and lab work that I talked about, we've also been doing quite a bit of work with stakeholders, and that dates back to 2013, when we met to kind of discuss what some of the issues are and what the research priorities were for the country were. There have been a number of workshops that have been held concurrently when the Bahamas Natural History Conferences have happened, where we talked about these issues, worked on a SWOT analysis, and discussed developing a framework that would outline and address all the research that we need, work on education and outreach and advocacy so that we can really promote sustainable management of the fishery, because that's important. We've also worked on stakeholder assessments to better understand perspectives of Nassau grouper as it relates to environmental change, fisheries management, legislation, enforcement, science, education, and outreach, and to assess initial or preliminary support for willingness to change or introduce fishery regulations. We also established a Nassau grouper working group, and we meet at least 4 times a year to work through some of these issues. And then the Department of Marine Resources also led a nationwide survey in 2019 and 2020 to get additional feedback from a broader range of stakeholders about management options. And this is what really fed into all this information, all this work. So the biological components, the work with stakeholders and getting that feedback is what led to the creation of the management plan for Nassau grouper. And we know from a conservation perspective, we also need to include and preserve genetic variation to mitigate biodiversity loss. And so integrating this into the management process is going to be really important moving forward. But for the management plan that we developed, the goal really is to promote population recovery of Nassau grouper because it is critically endangered, and sustainability of the grouper fishery, because it is an important commercial fishery species. So there are biological justifications for the recommendations that have been outlined in the plan. And in collaboration with the working group, we've been further advocating for changes through the submission of a policy brief that was also submitted to government back in 2018. Other implications then from this work mean that we're hoping to see some changes to the fishery regulations so that the size limits are adjusted, because what we currently have is too small. Whether or not there will be any changes to the duration of the closed season remains to be seen. The current closed season that we do have does sort of protect Nassau groupers during some of the peak months that we know of currently when fish are spawning, but it doesn't encompass the entire reproductive window for this species. We also think it's important to remind people of the restrictions on gear use. Often at some of these spawning aggregation sites, we see people out using hookahs, and these sites are really deep, so it exceeds the depth limit for using that type of equipment, and to remind people to use biodegradable fasteners on fish traps so that they don't end up engaged in ghost fishing, and considering implementation of other regulations as well, things like quotas. Enforcement has and is a challenge for the Bahamas given our scale and scope, and people can't be everywhere at the same time. We recognize that IUU fishing is a problem, and these threaten our food security, obviously have implications for biodiversity, but there are national security implications as well. But there are a lot of existing mechanisms that exist within the region to help governments and agencies with addressing IUU fishing. And I know there's been a lot of work on this level at the regional level, and the Department of Marine Resources has stepped up, and the Royal Bahamas Defense Force, their work on enforcement, but they could use more support. Obviously, in order to adaptively manage the fishery and figure out what's going on, if the regulations are being effective, we have to be able to do research and monitoring. Currently working with Daniel Pauly and his team on comprehensive stock assessments. But there are other threats to Nassau grouper, and it would be really valuable to do additional work looking at connectivity and how threats to their habitats may be impacting their populations, distribution, use, etc. But it's also really important that moving forward, we incorporate this information into our protected area network as well. And a few years ago, we developed a marine biophysical monitoring plan for the protected area system to help kind of guide next steps as a framework to guide next steps for monitoring within protected areas. So I think for the purposes of today, I think the take-home messages are this. We know not just here in the Bahamas, but throughout the region, Nassau grouper is extremely important for the environment and maintaining healthy reefs, but it's also been culturally and traditionally and economically important for people as well. Their life history makes them really susceptible to being overexploited. And FSA fishing in particular is not a sustainable practice. It undermines the ability of populations to adapt to stressors and their ability to replenish the fishery. So that's something that we would like to discourage. But on the positive side, we know that recovery is possible if fisheries are managed well. And there are examples of this in other countries. The Cayman Islands, for example, are seeing a recovery based on well-enforced fishery regulations, and their spawning aggregations are doing well, and they're seeing grouper on the reefs. So this is something that's doable, and we can do this here. And one of the things that you can do if you want to help is to respect the closed season. So that means not fishing for grouper, not buying grouper, not eating grouper, and not selling grouper during the established closed season for the Bahamas, which is December 1st through February 28th. Please leave the juveniles, allow them to reproduce at least once. before they're caught. And because this is a critically endangered species, I know it's harder for fish, but you know, I would encourage people to try and reduce their consumption of Nassau grouper. We have this year, we'll be launching our sustainable seafood campaign, and I think that will provide people with other alternatives to Nassau grouper that you can eat during the time when the season is closed. One of the other things that you can do that would be really, really helpful is to go to our website. You can scan this QR code. And if you are familiar with spawning aggregations, you can help by reporting sightings to us here, as well as donating to our fisheries program. That would be extremely helpful. The last thing I want to say is that we have been working regionally with CAFC's spawning aggregations working group to help elevate awareness about the importance of fish spawning aggregations and the value that they bring to the region. And for those of you that aren't familiar, Big Fish, it's a trilingual, so an English, Spanish, and French campaign. You can follow them and find them on social media and LinkedIn to learn more. And we've been collaborating with Big Fish on some materials. If for those of you that listen to local radio, you may have heard one of the radio PSAs this month has been playing for the month of February. But there are a number of other products like short films, and we had a special screening of one of the long films thanks to Bahamian last month. So please do learn more about Big Fish and how you can help. There have been so many people involved with Nassau grouper research and conservation and advocacy and education over the last 10 years, longer even. I started doing this work back in 2010, but there are people that have been at it much longer than that. So I want to acknowledge them and thank everybody that's been involved in various aspects of this work, whether it's the research, whether it's the survey work, whether it's the education, outreach. We really appreciate your contributions and to all the funders as well. Here is my contact information. If people have any questions, if you want to talk about things that are a little bit more technical, you can shoot me an email. I'm also on social media. So with that, I will stop and turn it back over to Danielle to address, I guess, any questions that the audience may have.
Thank you so much, Krista. That was an awesome presentation, really informative and detailed. So before we get into the questions, I just want to bring the audience's attention to the chat. There has been a link shared for the webinar. It's a feedback survey. So during this Q&A session, if you, the participants, can fill out that form for us, we would greatly appreciate it. So I'm just going to get started with the questions. So first question is, Charlotte wants to know, are there any plans to get Nassau grouper off the menu during the off season? It's hard to prove it was taken before the season was closed, same as for lobster.
Yes, that is very true because that's something that has constantly come up. And I don't know how to answer that question because at this point that's kind of voluntary. So yeah, I don't know what to tell you there, Charlotte.
Okay, next question is from Steve. What is the depth range for the fish spawning aggregations? Is there any possibility that there are unknown FSAs that occur at depths below what standard dive ranges would allow to be discovered?
Yes, that's a really good question. For us, because we're not doing any technical diving, it's no decompression diving. We don't do anything deeper than 140 feet. And obviously, even at that depth, we have a limited bottom time. So typically when we're doing these surveys, we try and stay between 80 and 100 feet in depth. But fish are definitely below that. And it is definitely possible that some of these spawning aggregation sites are occurring below the depths that we can actually do assessments, and that's why it's so important to be able to use some of these other techniques to monitor what's happening. It would be nice if we were able to do more of that. In some areas where people have more resources, they've been able to do things off of submersibles. There's the acoustic, the other type of acoustic work, the passive acoustic work that I mentioned briefly, where you can put down a recorder, and that can be used to capture the sounds that groupers produce at these spawning aggregation sites. And then you can use that to kind of verify presence, absence, along with deploying video instruments as well. So those techniques do exist, and that is definitely a possibility. So thanks for that question.
Awesome. Okay, next question is from Michelle. Do you think that the recovery of the species requires a human-assisted input of genetic diversity, given the low numbers of spawners at traditional FSAs?
That's a good question. And I'm not sure what the answer is. I think what would be more useful what we should try and do at this point is really discourage fishing at these spawning aggregation sites and see, how much recovery happens on its own. It's something that we know can happen within a 10-year time frame. It's just the persistent fishing of the spawning aggregation sites is likely to have some implications for the genetic health, and we're seeing that already long term with this fish. Whether or not we should be experimenting and altering that, I don't know. It's not something I would recommend at this time. But I know there has been some interest in restocking initiatives, but you need to worry about things like inbreeding as well. So just something to consider.
Alrighty. Next question is from Marsha. Is your work affected by the denial of research permits in the Bahamas? If so, how?
Yeah. It has been challenging the last several years to get permits in a timely manner to study this phenomenon. If we get it after spawning season's already ended, that's not helpful. I did get a permit for this current spawning season, but the weather did not cooperate for the areas I was permitted to do work. So, I'll have to reapply and try again for next spawning season. But the permitting situation has definitely, unfortunately, impacted some of the work that I'd been doing and plan to continue doing in country.
All right, thank you for that. Before I move on to the next question, I just want to remind the audience that her information, Krista's information is still on the screen. So if you have any questions or any requests for any documents or links or whatnot, you can always reach out to her for those. So next question is from Ramon. Is there any partnership with fisheries for artificial enhancement of juvenile populations through aquaculture?
No, not at the moment. For a while there was a Tropic Seafood had been doing aquaculture with Nassau grouper. Their challenge was I talked about it briefly, but mortality is really high in that initial stage, when then that larval stage. And that was something that they struggled with. Aquaculture initiatives for grouper have been a little bit more successful in other countries like Asia, but they're usually using that to directly generate product to go for food and not for restocking purposes. So it's expensive to do that, the capital cost of getting things start up, and there has to be a return on investment for that to continue.
Okay, understood. Next question is from Avisha. I'm so sorry if I'm pronouncing your name incorrectly. What advice would you give to persons who aren't involved in the marine field outside of donating and not ordering grouper that can assist in the cause?
Yeah, that's a good question. Even if you're not a scientist and you don't eat fish, there are other things that you can do. Nassau grouper, we didn't spend too much detail, but they use a range of habitats. And for anybody that loves to be out on the water, in the water, making sure you're doing your part to keep other ecosystems that they need in order to survive healthy would be really useful. So avoiding anchoring on reefs and damaging reefs. We know stony coral tissue loss disease is something that exists now that's impacting the diversity and composition of reefs, and that three-dimensional structure, those reef-building corals are really important for providing habitat for species like Nassau grouper. So disinfecting gear if you're in the water and moving around in places where you know that disease exists would be good as well. Obviously, not polluting is really important, and trying to make sure that we have good water quality. Those are things, but just simply talking about these issues. If you see something, say something. Educating other people about the importance, why we have a closed season and how important that is for this species long term to survive and to replenish the fishery and to keep going. All of those things are things that you can do to help.
Okay, thank you for that. We have a comment from Kasarina. This is in response to the first question. The legislation prohibits sale and purchase of Nassau grouper during the closed season. This is different from the crawfish legislation that refers to fresh crawfish. I just wanted to make that statement because it wasn't made to the actual group and it is in response to a question. So, Krista, do you have anything to say to that before I move on
No,
to the
that is
next
what the
question?
legislation, yeah, that is what the legislation says. However, restaurants and fishers have been allowed to sell product if they're inspected prior to the start of the closed season. And so people can say that it was collected before the closed season. So unless we work around that, I think that's something that's going to continue to be a problem.
Okay.
Yep.
The next question is from Francisco. How can the community get involved in education as well as helping the authorities?
A lot of there are a lot of organizations that have, aside from us at PIMS, have materials and opportunities for people to get involved with research and outreach with restoration. So those are ways that you can get involved um, and helping with this issue. I forget
Alrighty,
the
next
last part of your
question.
question, Danielle, sorry.
No, it's just how can you get the community involved in education as well as assisting the authorities?
Yeah. So the biggest thing would be to, depending on where you are, getting in contact with some of the local NGOs or environmental groups that are doing work. They often have a lot of resources that are available and then can put you in contact with the right people. So that would be my recommendation there.
Okay, Laura has a question. How long do juvenile fish spend in the mangroves as she thinks it's very important for their reproduction?
Yeah, that's a really great question. So they're going to be in those sort of nursery habitats for about the first 10 months of their life or so, before they start making the transition to these patch reefs where they can, or like hard bottom habitats where they can potentially find like nooks and crevices to tuck in and settle. So that first 10 months is really critical, and that's when they're in those nursery environments.
Alrighty, next question is from George. Do you know of grouper FSAs within marine protected areas, either within or outside your study area in the Caribbean?
Yes, is the short answer to that question. Even within one of the slides that I talked about, there are, for example, in Exuma Keys, there are there's spawning aggregation sites, sorry, within the protected area, but for whatever reason, the fish decide they want to go somewhere else to breed. So they do exist.
Okay, cool. I don't see any more questions. Are there any more questions for our speaker today? Just going to give you all a few more seconds to. Okay, next question is from Lester. Do you think climate change will affect spawning with not with the Nassau grouper?
Hi, Lester. Yeah, that's a really good question. There's been some modeling work done to look at that and how that may impact the timing of migrations to and from some of these spawning sites. Um, it often occurs in the winter months when it's cooler, and as we see increasing temperatures in the water, that may likely shift the timing of when these fish are making those spawning migrations and participating in spawning events. So the timing of reproduction, that window may shift. The other thing is we don't really know how that's going to impact developmental stages, the you know of a fish in the early developmental stages, so the larvae of Nassau grouper. So it's a possibility. I know there's been some experimental work looking at how impacts on physiology, but there's a lot more work that needs to be done in terms of looking at climate impacts to the species themselves and how that may influence and impair reproduction and the timing of these spawning events and what we then need to do to adapt our regulations and policies to reflect that.
Alrighty, thank you for that. I'm just going to give the audience one more chance to insert any more questions that they may have while I remind you all of the feedback link in the chat. That's for this webinar. So one second, I think we have another question.
Sure, no problem.
Oh, it's just Valeria saying thank you for this amazing webinar.
De nada, Valeria.
Okay, I think that is all for now. But as I mentioned, if you all have any questions for Krista, you can reach out to her via her contact information on the screen. I would recommend that you all record it in some way, especially if you may have any sort of questions. As well, a final reminder to please fill out the feedback form. The link is in the chat. Um, in closing, thank you all for joining us today to learn more about the Nassau grouper, the heart of Bahamian fisheries. Thank you again, Krista, for this wonderful presentation. It was really informative. For more information, please visit our website at perryinstitute.org. Follow us on our social media channels at Perry Institute for Marine Science, including Facebook, Instagram, Twitter, and LinkedIn. We hope you will join us again for our future webinars. Thank you again so much for attending, and please enjoy the rest of your day. Bye.
Thank you, everybody. Bye.